folks. I gotta tell you, I'm surprised I don't hear more crickets here today. I was expecting you guys knew something was up on Thursday. There are no classes tomorrow. Don't come to any classes you have tomorrow. But good to see you all. I think we can make this pretty fun and relatively short. It seems to me every time I promise to hold a class out early, though, I don't deliver on that, so don't hold me to it. But I want to talk with you today about one of the things, one of the coolest things that I know of in the whole area of biology, and that is how it is that genes get turned on and turned off. There's a whole class, a whole semester-long class about this, Bio 2120, that back when we were on quarters, I was the only one who ever taught. But now Scott Baird in the biology department often teaches that class. Absolute best class ever. Get to really get down and into the nitty-gritty about how bacteria turn genes on and off. Amazing stuff. I'm going to give you a taste of that today. Just before we get into that, though, let me pick up right where I left off on Thursday of last week. You'll remember on Thursday of last week, I just started to talk with you. I sort of waved my hands at the idea of viruses. I can tell you just a bit more about viruses. Maybe we'll wait until the traffic to clear a little bit. So viruses are really simple, right? They have, most viruses just come with what they need to be able to take over the metabolic machinery of another cell, or of a cell, copy themselves, and then off they go. But they all have a nucleic acid of one kind or another to store their genetic information. And many viruses now, hundreds of viruses, have been completely characterized at that level. We know every letter, every nucleotide that spells out their genetic blueprint, right? And so people have been able to study them and look very carefully at how it is that they get their genes expressed and how it is that they manipulate the gene expression system of the cells that they infect. So we've learned a lot about gene expression from the study and the characterization of viruses, and a lot of that is going to influence it's going to influence the things that I talk with you about later today. So I talked a little bit about viruses on Thursday. I said a few more words about them today. And on Thursday, I left you hanging with this idea of conjugational mapping. And I really just sort of lobbed it out there. I left a question hanging on the screen as we wrapped up class, and I said, here, have fun with this. You can probably figure it out without my help. So let's see if you guys did that. If you didn't, I'll take you by the hand and we'll talk about how conjugational mapping works. So here's the deal. That's a map of a bacterial chromosome. My recollection is that's sort of superficial, sort of high-level map of E. coli's genome. E. coli's genome, it's a 4 million nucleotide long circle, G's, A's, T's, and C's all the way around, and it spells out all the instructions that E. coli needs to be a happy, free living organism. It doesn't really need help from anybody. It can take care of all the things that it needs. It can make all its own amino acids. It can make all of its own sugars, lipids, anything that it needs, it can take care of by itself. The instructions for that are spelled out in a 4 million nucleotide long chromosome. And again, this is a map of that chromosome as a bacterial geneticist would present such a map. What you'll notice, things that jump out at you, is, check it out, there's a stretch of 4 letters here, A, B, C, D, and they're attached to Lu. I'll tell you, just as a for instance, what they're telling you there is that that's the leucine operon, L-E-U, that's the 3-letter abbreviation for one of the 20 amino acids, leucine. And those A, B, C, D above it, those are telling me, I've done no prep work for this. I'm doing this, at least for this part of the lecture, I'm doing this just from looking at that map. Apparently, there's a series of 4 genes that are involved with leucine synthesis. Right? So there's an operon, I can tell that from where the arrow is pointing, my cursor arrow. That's the leucine operon, and there are 4 genes there, A, B, C, and D, and I can tell you from experience and just from knowledge of how these bacterial genomes get put together, those 4 genes are going to be playing an important role in the biosynthesis of the amino acid leucine. And so you're looking at this and saying, gee, what do I need to know? Am I going to be asked to? It's the same for all of them. And I'm going to give you a specific example later. Let's see if I can find it on there. Yeah, there it is. This is the tryptophan operon over here. So elsewhere on the bacterial chromosome, I think, is the main point for you to take away at this stage. But somewhere else on that E. coli chromosome is something called the tryptophan operon, TRP, 3-letter abbreviation for the amino acid tryptophan. There are 5 genes that need to be expressed in order for bacteria to be able to synthesize the amino acid tryptophan. They're all clustered together. They're arranged A, B, C, D, and E. Right? Those are the 5 names for the 5 different genes that are involved with the synthesis of tryptophan. And so what you could take away from a sort of a first look at a map of a bacterial genome like this, and as I'm looking at it now, it seems to me what they're trying to do with this map is show you where all the genes involved with amino acid biosynthesis are. There's obviously many other genes involved with glycolysis and ribosomes and DNA polymerases. Those aren't shown on this map. They're just showing you where the genes that are involved with making amino acids are. Notice that they're often clustered together in these operons. That's what an operon is. It's a cluster of genes. And bacteria tend to be, for lack of a better phrase, kind of anal retentive about the way that they organize their genomes. Genes with related functions are often right next to each other, very neatly, nicely organized. There's nothing at all like that in eukaryotic genomes. We've got a gene that codes for alpha globin, a gene that codes for beta globin. You need to make both those proteins to be able to make hemoglobin, to carry oxygen in our blood. They're not just not next to each other, they're on separate chromosomes. Right? We don't organize our genes the way the bacteria do. The bacteria have this tendency, very often you'll find genes with related function are clustered right next to each other. And you'll see, as I go through today's lecture, what a help that is to them, to bacteria, in terms of coordinating their gene expression. There's no point in making the first gene, or making the protein coded for by the first gene in tryptophan biosynthesis, unless you're also going to be making the second gene's protein, right? 1/5 of the way to tryptophan isn't doing anybody any good. You got to make sure you make all 5 of those proteins to be able to get the job done. You have to coordinate the expression of genes. Bacteria have a simple, easy way of coordinating the expression. They cluster them all together, and they get them all expressed at the same time. We've got a whole lot more complicated, different, and less efficient system than what bacteria have. So again, all that you really should be looking at this map here is that, hey, check it out. People map out genes on these circular bacterial chromosomes. They have a sense of where the leucine operon is relative to the lactose operon, relative to the galactose operon, relative to the tryptophan operon. Right? They're all laid out in a particular order, and people have characterized where those are. The way that they get characterized, at least if you don't want to roll up your sleeves and do DNA sequencing, frankly, that's the way that typically you map out a genome these days, is it's come to the point now where it's actually easier to sequence a genome than to do conjugational mapping. That's turned the world upside down from the perspective of many microbial geneticists, where only up until about 5 years ago, sequencing a genome of a bacteria was a $1 per base pair kind of proposition. 3-4 million nucleotide long genome cost about $4 million to sequence. So you can put a graduate student to work on doing conjugational mapping for a semester, cost you about $10,000, or you could pay $4 million to have the genome sequenced. It's a pretty easy decision to make. Now genome sequencing is coming to the point where it's competitive with graduate student time, and so again, the world's changing. But the way that most bacterial genomes, up until a few years ago, were being mapped out was through conjugational mapping. And I think you guys will get a kick out of how it is that this conjugational mapping works. I don't think it's come up in this class. I remember talking about yeast mating types. I don't think we've talked about E. coli having sex, right? Everything seems to have sex, right? You got E. coli, which is capable of having sex. That's what conjugational mapping means. You're interrupting E. coli in the midst of having sex. The way that bacterial sex, and specifically the way that E. coli sex works out is this, is there is an extra chromosomal circular bit of DNA, okay, called the F plasmid. A plasmid is, in bacterial parlance, when we're talking about bacteria, when we're talking about prokaryotes, a plasmid is a circular bit of DNA that's separate from the chromosome. They tend to be very small, usually a couple thousand nucleotides long, as opposed to a couple million nucleotides long. Many bacteria have plasmids, and in fact, many bacteria have many plasmids. The reason that you get antibiotic resistance spreading in bacterial populations is typically because the genes responsible for the metabolism of what used to be a toxic substance to the bacteria, those genes reside on plasmids, and it's pretty easy to transfer a plasmid from one bacteria to another bacteria. Okay? But here, for conjugational mapping, we need to just focus on one kind of plasmid. It has nothing at all to do with antibiotic resistance, aside from the fact that it's on a plasmid like those types of genes tend to be. And the specific plasmid we want to talk about is one called the F plasmid. Here they're calling it the F factor. And what the F is short for is fertility. An E. coli that has an F plasmid has on that plasmid a set of genes that allows the E. coli to make what's called an F pilus. It's a protein structure that pokes out from the surface of the bacterial cell. Okay? An F plasmid lets you make an F pilus. Bacteria with F pili, just the plural for pilus, a bacteria like E. coli with F pili poking out of its surface is set up, it's ready to go to poke into other E. coli, particularly ones that don't have F pili. All right? It's kind of a male-female kind of thing going on here. And so the E. coli with the F pili can poke into E. coli that don't have an F pili, and when they do, what happens is the F pilus, there's proteins that are coded for by the F plasmid that cause the F plasmid itself to get replicated in sort of a rolling circle kind of model, sort of a rolling circle approach, and the F pilus gets snaked through, the F plasmid gets snaked through the F pilus from the one cell to the other. So you make a copy of that F plasmid and you transmit it over into the cells that didn't have an F pilus. All right? Simple, right? Nothing too big. If it sounds complicated, it's just because I'm doing a really bad job explaining it, right? The picture kind of says it all here. There's two ways you can get cells with F pili. You can take an existing cell with an F factor and replicate its chromosome and its plasmid the old-fashioned way, and where you had one cell, you get two. Here's the other way. It's the conjugational way. Here, those squiggly things are F pili. The squiggly things let you have one bacteria, one E. coli, hook up with another bacteria that doesn't have the squiggly things, and the F plasmid gets copied. The DNA polymerase goes around in a circle, and the F pilus facilitates the transfer. It's a hollow tube through which the DNA gets of the F plasmid. That copy gets snaked into the other bacteria, and you end up then with two cells that have F pili. All right? So what does this have to do with conjugational mapping? So far, we're at the conjugational part. Right? That's the sex part. Here's where the mapping part comes in. It's possible to construct E. coli cells where the F plasmid isn't a free-floating little circle, but rather the F plasmid is incorporated entirely into the E. coli chromosome. Okay? E. coli's got that big circular chromosome, got the little F plasmid. If you cut the F plasmid and make it linear, and you cut the chromosome and make it linear, and then stick the F plasmid into the linear version of the chromosome of E. coli and recircularize it, you've got an E. coli chromosome that has an F plasmid integrated into the E. coli chromosome. And that cell still has all the genes that you need to make an F pilus, right? And it has all the stuff that you need to initiate a replication of that F plasmid. But then what happens is after you've copied the F plasmid, the DNA polymerase just keeps running right along, and now it's making a copy of the E. coli genes. And those are the things that get snaked over through the F pilus into the receiving E. coli cell. Okay? The longer that you let that conjugation play out, the longer you let that DNA polymerase go around the E. coli chromosome, not rocket science in any sense, the more genes get transferred over. And if you interrupt this conjugation, if you shake the cells up and you separate them effectively, you interrupt the transfer of DNA from the cell that has the F plasmid in its chromosome into the next cell, and you interrupt it, right? So you can control how long or how much of that E. coli, the first E. coli's DNA, gets copied over into the other E. coli cell. The whole process to copy the entire E. coli chromosome in this kind of conjugational mapping takes somewhere between 45 and 60 minutes. And so if you stop the process at the beginning, you might only get, so here's where the F plasmid is integrated. They have it shown here as blue, right? At the start, you start sneaking stuff through there, and then you replicate through, and you get all the way past A, which is part of the E. coli chromosome, into B, and A now has made it into this other cell. After A, you replicate through B and then get over to C, and B and A make it through to this cell. Right? If you were to break that bridge at this stage, that's all that's going to get over. It's going to be the first couple genes after where the F plasmid integrated into the chromosome. But if you let it go another half hour, you may get the whole E. coli chromosome transferred over. And so what happens with conjugation mapping is really simply this. You set up a chromosome with F plasmids integrated into it at different positions, maybe 5 or 6 different positions. Okay? So you set it up, you make your E. coli, you get the F plasmids into 5 or 6 different spots along the E. coli chromosome, and then you let those E. coli conjugate with other E. coli, and you look to see the order in which the genes get transferred from the one cell into the other cell. And what you get at the end of that sort of experiment is this kind of data. So this is the kind of data you would expect to see in conjugation mapping. So an E. coli that has the F plasmid integrated into its chromosome gets a fancy name. It's called an HFR, high frequency of conjugation. That's what that's short for. But you make an HFR strain. That means you've put the F plasmid into the bacterial chromosome. And you can see in this example we've got 5 different HFR strains. Nothing magic to their names. They're arbitrary names, H4, 6, AB311, AB313. I'll bet you AB are the first initials of the graduate student who made these. That's the way it usually works. Right? And so you've got your 5 different integrations of the F plasmid into the circular E. coli chromosome. And the first thing that always gets transferred over is the F plasmid itself. That's what the O is for. It's the origin of replication from the F plasmid. But then Notice for this strain, the next thing, the very first thing really that you see getting transferred over is a gene that's involved with threonine biosynthesis. And what it does isn't important. It's just, look, there's a cluster of genes, and we'll call it THR. Then there's another cluster of genes, then another cluster of genes, and another, and this is the order in which these clusters of genes are getting transmitted from that HFR strain into other bacteria. And so that tells you something about the order in which these genes are arranged, right? At one point on the chromosome, there's the threonine genes, the threonine operon. A little bit further along is the TON operon, and a little bit further away still is the lactose operon. We've mapped out the order of some genes. Kind of cool. That could be useful for certain experiments. If you wanted to manipulate these bacteria, it might help you to know the lay of the land along the length of the chromosome. So one HFR strain gives you a picture of a portion of E. coli, or whatever bacteria you're working with, lay of the land, where the genes map out along the length of their chromosome. And a different HFR strain might give you a picture of a different part of the chromosome. Because again, remember, the F plasmid here will have been integrated into a different spot. And now we want to see what genes get transferred over and in what order from that other spot. So you start at a different place on the circle of the E. coli chromosome, and you look to see what genes come over. Now, as I look at the genes that came over for 4 relative to H, I don't see any overlap or similarity between them. So what we're getting here is a look at 2 different stretches of genes along that 4 million nucleotide long chromosome. I don't know their orientation relative to each other. I don't know how close or far apart these sets of genes are, but I know for these little patches, that's the order in which genes are playing out. For the 6th strain, and not, again, F plasmid integrated in a whole different spot along the chromosome, I get another set of genes that tells me that first you get this cluster of genes, then this one, and now for the first time in this example, I'm seeing some similarity. I've seen that cluster before. ILE here is the same as ILE there, right? MET here is the same as MET here. THI here is the same as THI here. They're in different order. You know what's going on? Is the F plasmid, in one instance, got integrated in this orientation, and in the other instance, it got integrated in this orientation. And one of them is pointing this way, along the circular chromosome, and the other is pointing this way along the circular chromosome, and they overlap. We get those 3 clusters of genes, one in this order, one in this order, but then, if I wondered what was on the other side of my thumb going around the circle, I can look at the information I got from the 4 strain to tell me what comes before. And if I wonder what's going on the other side of this thumb, all I need to do is take the information off of the 6th strain, right? And so now I've got some overlap, and I can start to make a broader map, not just from one stretch, but from 2 stretches because they're overlapping with each other. And I can start now to make a map that might take me all the way around the whole circle. And again, over here, we've got a different strain, and you can see this is the sequence of genes that come out. Hey, look, there's some overlap there, right? And so I can tell you that what comes next in line, you go LU, AZI, TON, and then PRO, LAC, ADE, GAL, TRP, HIS. Right? See how that goes? The ton overlaps. The PRO must be the thing that's on the other side of the ton, right? And I can now string these things together. And remember, this one overlapped a little bit with this one. Those 3 overlap with these 3. Then I'm starting to patch my way around the whole circle. And actually, when I look over here, I see LAC here and here, ADE here and here. I think I've made the whole circle now, or at least I'm very close. It's possible I'll need this one. So that's it. That's the sort of puzzle. It's the molecular biology or the molecular microbial genetics equivalent of Sudoku, right? You get 4 or 5 pieces of information, and from it you're able to fill in that whole circle. And you can make a map of the entire E. coli chromosome. So I told you at the end of class on Thursday that this is a question that I think I've had on every exam that I've ever given to Bio 1120, right? And I promised you at the end of class on Thursday I'm going to give you a question just like it. So I'm hoping that you can just take a few minutes, if you're unsure what I've been talking about right now, just sit down with a paper and pencil maybe and sketch things out. I think you might find it's a kind of interesting puzzle to solve. I like you to be able to solve that puzzle because if you can solve that puzzle, in a nutshell, you know how conjugation mapping works. Right? And conjugation mapping has been a big deal in the history of mapping out where genes are on bacterial chromosomes. And so what you'll find is only one of these 5 corresponds to something that you would get off of that circle. And what I'll probably do for you guys is go, scrap these and come up with a different set of genes off of that same circle to give you as the choices. So all you need to do is be able to make the circle. It'll take you on the exam. It'll take you about 1 minute, maybe 2 if you're a slow worker. And then you can just figure out which of those actually corresponds, which of the choices corresponds to the order of genes that you might see on that circle. All right? If you're wondering what's going on, try it. You'll be amazed. You'll have fun. You'll share it with your friends. I've given you a gift that you can share with your family around the Thanksgiving table on Thursday. Right? Everybody loves conjugation mapping. If you got young kids in the room, leave out the conjugation part. Right? Make it G-rated. But the mapping part, grandmothers, everybody loves to do mapping experiments. All right, so there you go, conjugational mapping. Here's the actual really good part. I want to talk with you about the regulation of gene expression. Now let me give you a setup to this before I tell you the nuts and bolts of what's going on. I've said it a couple of times to you. All right? I want to really emphasize it today, perhaps more than ever. It's a each bacteria world out there. And what causes one bacteria to fare better than another, ultimately, almost always comes down to how efficiently they can use their energy. Right? How efficiently they can capture energy, how good they are at taking energy from their environment, how good they are at storing that energy, how good they are at using that energy, being efficient, not wasteful, only burning ATPs and breaking down glucoses when they need to. Right? That, more than anything, governs, it drives, it dictates everything that bacteria do. It's job number 1. Save energy. Energy is precious. The better a bacteria is at saving and hoarding its energy compared to its competitors, the better it's going to do. Pretty much nothing else matters. For a bacteria, for a prokaryotic organism, energy, efficiency, job number 1. I've said it now for 30 seconds, 6 different ways. I can't emphasize it enough. It is the overriding concern from a prokaryotic organism's perspective. Don't be stupid with your energy resources. Okay? And what that translates to in the context of gene expression is really very directly this. Don't spend, I've talked about ATP as like a dollar bill equivalent, right? Don't spend your money, don't spend your ATP unless you're going to get a return on that investment. Every time E. coli spends an ATP at some level, a calculation is being made. Is this going to save us an ATP? Is it going to help us make another ATP? If it doesn't help you save an ATP, and if it doesn't help you make an ATP, don't do it. Period. And so bacteria need to know something about their environment, and they need to express their genes. And gene expression is shorthand. That's a buzz phrase that encompasses all of transcription and translation. For our purposes, we will say, I will tell you a gene has been expressed when a protein has been made. Right? When you make a protein, That's it. There's no equivocation, no quibbling. You expressed a gene. Did you just make the RNA? I don't know. I don't know if I want to call that expressed. You were starting to express, but you didn't finish the job. If you made the protein, you expressed it, right? And remember, you only make proteins if the making of that protein is either going to save you ATP or help you make ATP that you wouldn't have had otherwise. Anything else, a bad deal. Let me get you a sense of the scale that we're talking about here, the cost associated with gene expression. Stop and think. Let's think about what happens when you express a gene. First, you got to make an RNA copy of that gene, right? That's what the whole central dogma is about, right? Transcription, translation, all that stuff. You got to make an RNA copy. What does it cost to make an RNA copy of a gene? I talked a little bit on Thursday about a typical gene in a prokaryote. Let's say a typical gene codes for a protein that's 300 amino acids long. Okay? If it's 300 amino acids long, that means it's at least 900 nucleotides long. 3 nucleotides, triplet codons for each amino acid, right? So it's at least 900, and there's going to be stuff at the beginning, the untranslated region. Let's just be, let's make the math easy on me here, right? Let's say 1,000. 1,000 nucleotide long transcript. 1,000 nucleotide RNA has to be made if you want to make a protein, right? Plus or minus a couple. Not a big deal. 1,000 nucleotide long. How much energy does it cost to make 1,000 nucleotide long RNA molecule? How much energy, how many ATPs do you have to burn? Well, it's effectively 1 ATP for every nucleotide. Because one of the 4 nucleotides is ATP, right? So every time you integrate an ATP into an mRNA, that's an ATP gone. Every time you integrate a U into an RNA molecule, that's it. You got that from UTP, which is the equivalent energetically to an ATP, right? So just to make the RNA molecule, it's going to cost a bacterial cell on the order of 1,000 ATPs, right? In dollars, $1,000, that gets your attention, right? Like, what do you think? Do you want to go out to dinner tonight? It's going to cost you $1,000. Oh, okay, no, I'm not that interested in going out to dinner. I was in New York City yesterday. Seriously, yesterday. I feel like I'm still there in some sense. There's a restaurant that I walked past that's advertising Thanksgiving dinner, a $45,000 Thanksgiving dinner. It's like, I'm not into that. No, I don't need that, right? But I was curious. How do you make a dinner, a turkey dinner that costs $45,000? Part of it is that the stuffing includes a 2-carat diamond ring. That's not a Thanksgiving dinner. I don't know. That's not the kind of Thanksgiving dinner we have at my house. We don't put rings in the stuffing. But so nonetheless, 1,000 ATPs to make the RNA, right? And you haven't made the protein yet. How much does it cost to translate that RNA? Right? Well, for every amino acid that gets linked together by a ribosome, the effective cost is 2 ATPs, right? One to make the bond between the peptidyl bond between the 2 amino acids, right? And one to bring the tRNA in and kick it out when you're done. So we're up to, 300 amino acids times 2, 600 ATPs. We've got 1,600 ATPs to make the protein. And I don't know, I think we also need to take into consideration that when you've used those amino acids, they're not available for use to make other proteins, so there's a cost to making the amino acids. Each amino acid costs, depending on the amino acid you're talking about, between 12 and 72 ATPs, right? Let's just be friendly and call it 50, right? Somewhere in the middle. 50 times 300 is a big number, 15,000? No, 1,500 ATPs. Oh, it's 15,000 ATPs. Plus the 1,600 that we had made for the transcription and for the translation. We're talking in the ballpark of 20,000 ATPs to make one protein. Right? 20,000 ATPs before a bacteria like E. coli decides to make a protein, it's got to be confident that it's either going to save 20,000 ATPs because that protein's on deck, or it's going to make at least 20,000 ATPs because that protein's available to help with harvesting energy from something. It's a big deal. If an E. coli just frivolously makes a protein, I feel like making a protein today, no particular reason, it's at a 20,000 ATP disadvantage relative to all the rest of its competitors for every time it's frivolous like that. All right? That's the and again, bacteria, that's what helps them win in the battle against each other, is how much energy, how quickly they can bring their resources to bear. So here's the thing. It's a big deal. When an E. coli or any other bacteria decides to make a protein, it doesn't embark upon that frivolously or lightly. There's got to be a serious expectation of a return on that investment. All right? Big deal. And so, how does E. coli do that? Well, I've got up here a map for you of an operon. And actually, it's a map of a specific operon, though the picture doesn't give you the name of the operon. Yeah, I don't know, maybe it's not, the names of the genes aren't quite right, but the concept is the same. Here's an operon, all right? I'll give you a specific operon in a minute. Here's an operon. Here's the deal, all right? What we got is a set of genes. And again, in operons, we expect the genes to have related functions. So they're going to be doing something that's part of a process. Okay? And all three of these genes, they're located here, cleverly named A, B, and C. All three of them are located right next to each other, head to tail, along the length of the chromosome, 5' to 3'. And all three of them share a single promoter. And what that means is this. There's one spot that RNA polymerase scans along and looks for and says, hey, this is the start of a gene. This is a promoter. How does RNA polymerase know this is a promoter? Well, it's looking for specific strings of nucleotides along the length of the DNA molecule. 6 of them clustered together at -35, 6 of them clustered together at about -10, and 1 of them at the +1 position. Okay? String of 6 letters, 1 letter. When RNA polymerase comes upon that particular string of characters in that orientation, with that spacing between them, a little light bulb goes on, and the RNA polymerase says, ooh, that's what I've been looking for. And it sits down and it says, I think I might want to start transcription here, specifically at the +1 nucleotide. So that's how that magic gets worked out. The RNA polymerase is tasked with scanning along the circular chromosomes, left and right, looking around all the time to see if it can find those sequences. They're called the promoter. Okay? So the RNA polymerase finds the beginning of a gene at the promoter, and if it initiates transcription, if it starts to transcribe that gene, it goes through the whole operon from the beginning, the 5' end, to the 3' end. And as it goes, it makes an RNA copy of all 3 of those genes, A, B, C, right next to each other, all at the same time. If it makes an RNA for A, it's guaranteed to make an RNA for B and for C, and that's going to result in equivalent concentrations of the A protein, the B protein, and the C protein. In a word, that's coordinated expression, right? Beautiful, simple system. Why would you make one protein in the process if you're not going to make the other 2? Okay? That's how the problem gets solved for E. coli. But there's one last piece of the puzzle here. Right? So the RNA polymerase likes the promoter. The RNA polymerase really likes that, and we coordinate the expression by having all 3 genes get transcribed as part of a single RNA molecule. Incidentally, an RNA molecule that has more than one protein coding sequence within it is called a polycistronic messenger RNA. Polycistronic occurs only in prokaryotic organisms. All of our RNA molecules are monocistronic. We never have more than one protein spelled out along the length of an RNA. So, but in prokaryotes, polycistronic messages are very common. All right? So you got your polycistronic message. There's one last piece of the puzzle. It's this word operator. And the operator is a wondrous thing. The operator is where some other protein can bind. RNA polymerase binds to the promoter sequence at -35, -10, and +1. Nearby, for most prokaryotic operons will be another site that another protein can bind. And that other protein is going to be a regulatory protein. So specific string of nucleotides that this protein looks for, different from the -35 and the -10, but some other protein can recognize and bind these sequences. And let me show you how this plays out. Here's a specific example for you. There's an operon, and this is the first operon in E. coli for which this got sorted out. Okay? These guys got a Nobel Prize for sorting this out. It's the lactose operon. You guys have heard of lactose intolerance, that sort of thing. Lactose is a sugar. It's a disaccharide, abundant in milk. Lactation, lactose, it's milk sugar is what it comes down to. It's a disaccharide. And like any disaccharide, I hope one of the first things that comes to mind at this stage in this class, when I say sugar, when I say disaccharide, I hope you perk up and you say, ooh, energy. There's some energy there. That's certainly what an E. coli is thinking when it gets a whiff of a sugar. Oh, this is promising, right? I'm interested. Let's talk some more, right? It's a sugar. It's an energy source. If E. coli finds itself in an environment where there's some lactose, that E. coli is well served if it's able to capture that lactose from its environment and then break it down and get it shuttled down through glycolysis so that it can make ATP, right? And so if there's lactose in the environment, it's a good idea for E. coli to metabolize it in a single word. That'd be a good idea, okay? If there's no lactose in the bacteria's environment, there's no point in making the proteins involved with lactose metabolism, though. So it's an either-or sort of thing. If there's lactose present, express the genes of the lactose operon. If there is no lactose present, don't waste your time. More importantly, don't waste your energy. It's as simple as that. So what do you need to be able to metabolize lactose? Apparently all E. coli needs to metabolize lactose are 3 proteins. The first of which is Z. There's also a Y and an A. Jacob and Monod are the French guys who got a Nobel Prize for figuring that out. Just to stick it to them as Americans, we often refer to them as Jacob and Monod, right? But the French is Jacob and Monod. These 2 guys realized that you need to make these 3 proteins in equal amounts to be able to have a bacteria that metabolizes lactose. The first protein is the protein product of the LAC-Z gene. Okay? I'll give you some shorthand here. Just got to give you a scorecard so you can keep track of the players. When I'm talking about a gene, I'll just say LAC-Z. Right? That means the gene, the DNA sequence that codes for the protein that is made from that gene. That's a gene. LAC-Z means gene. If I say P-LAC-Z, if I put the letter P in front of LAC or the name of some gene, that means I'm talking about the protein product of that gene. Okay? The enzyme that gets coded for by the LAC-Z gene. P-LAC-Z has a simple job. Its job is to take the disaccharide lactose and break it into 2 monosaccharides that can get shunted through glycolysis. Okay? Its name is beta-galactosidase. That's the formal name for it. You don't need to worry about the name. PLAC-Z plays an obvious role, an obviously important role in the metabolism of lactose. It breaks it into two monosaccharides. It can be run through glycolysis. Clearly important. PLAC-Y, the next gene in the operon is LAC-Y. PLAC-Y is PLAC-Y is a protein that is membrane bound. It's in the E. coli's exterior membrane that is a pore, a channel through which lactose can move. Right? So lactose outside the cell can get to the inside of the cell through this particular pore, PLAC-Y. It's a diffusion thing. If there's more on the outside than the inside, it comes in. Okay? The last gene in the LAC operon is LAC-A. PLAC-A codes for a protein that modifies lactose in such a way that it can't get out the PLAC-Y pore. Kind of a clever trick, right? Once it comes into the cell, it could have gotten out, but it gets modified so that it can't get out through the pore anymore, and it's a one-way ticket, essentially. Once you come in, you get modified by PLAC-A, and you can't get out, and ultimately, once you're in, even after you've been modified, you get broken down by PLAC-Z, and you get shunted through glycolysis. So that's how lactose metabolism works. You get all three players. There's no point, again, in making PLAC-Z if you're not making PLAC-Y. There's no point in making PLAC-Y if you're not making PLAC-Z. You want to coordinate their expression. And you want, ultimately, here's the bottom line, you want to make those three proteins only if there's lactose outside the cell. That's it. That's the only time it makes sense to make that, because you're talking tens of thousands, probably hundreds of thousands of ATPs to make these proteins, and you don't want to do that unless there's a fair amount of lactose that you're going to be able to break down into ATPs so you can recoup that investment. So it really comes down to this. E. coli had better only express those genes if lactose is around. How is it going to figure out if lactose is around? It can't just have an open spigot and express these all the time, because lactose just isn't around all the time. Lactose, I don't know, maybe it's around 10% of the time. Tiny fraction. Expressing these genes when it's not around, bad idea. Not expressing these genes when it is around, bad idea, because somebody else will use that energy instead of you. You're losing, buddy. You better get on the game. How does E. coli pull this off? One of the things I talked with you about the first time we met is part and parcel with being alive is being able to respond to stimuli, to respond to your environment. This is where the rubber hits the road for a bacteria. How does it respond to the presence or the absence of lactose? It responds because ultimately there is a protein coded for by a different gene, the LAC-I gene. So there's 4 genes involved in lactose metabolism, the 3 that actually do the work, and the one that sort of controls the expression of the operon. There's a gene called LAC-I that codes for a protein called PLAC-I, or if you want, you can call it the LAC-I repressor. Right? But check out here, they've given you a shape of what PLAC-I looks like. It's not quite its shape, but notice how it can bind to the operator of the lactose operon. When the PLAC-I protein is bound to the operator, how does it know where it is again? It's looking for a specific string of nucleotides. It happens that that string of nucleotides occurs only once in the entire E. coli chromosome. It only occurs in the LAC operon. When PLAC-I binds to that string of nucleotides, it sets up a roadblock. It binds tightly, and it doesn't let RNA polymerase get by. In fact, it doesn't let RNA polymerase even initiate transcription. It can't even bring in that first nucleotide. So PLAC-I can turn off the lactose operon by binding to the operator. It simply sets up a roadblock. PLAC-I, though, is I don't know, you guys tell me, is it a 1 or a 2-trick pony? I want feedback on this, but it's not a 3-trick pony. I'm sure of that. You tell me how many tricks PLAC-I has, because it can do one or the other of two things. I think it's a 2-trick pony. One of its tricks is it can bind to the operator. The other trick is it can bind to, get this, lactose, right? But it can't do both tricks at the same time. I think that's a 2-trick pony. You guys with me on that? It's something. This is a protein that can do two things, but it can't do both at the same time. It does one or the other. It either binds to the operator and shuts down the expression of the lactose operon, or it binds to lactose and comes off the operator and allows RNA polymerase to transcribe on through. Problem solved. Boom. You know, I'm wearing a cordless microphone. I should have had the other. Just drop it and walk away. That's it. That's it. That's all. That's the whole show, folks. Oh, isn't this beautiful? At a molecular level, that's the appropriate response. At a molecular level, you guys now know how E. coli responds to its environment. Critical to being alive. And it's as simple and as elegant as that. One protein solves that whole problem. One protein. Yeah.
Does lactose get to the plaque-I? You mentioned that one of the proteins that plaque-I blocks is the channel that.
So this is an interesting question. If you haven't made PLAC-A, PLAC-Y, if you haven't made PLAC-Y to let lactose come from outside to inside, how does lactose ever tickle PLAC-I to come off the operator and allow expression to occur? The short answer, it's a two-part answer. The operator is a little bit leaky, and so sometimes PLAC-I isn't on the operator when it should be. It's asleep at the switch, and RNA polymerase can go through. But more likely and more importantly probably is that there's still some of that PLAC-Y protein in the cell's membrane from the last time the lactose was around, right? Not as much as when there's a full-blown lactose, flock out there, but there's still going to be some left over. So there's always a little bit of opportunity for lactose to come in, enough to tickle PLAC-I to get it off the operator and get the rest to work. So there it is. That's how E. coli regulates the expression of the lactose operon. It's the most important parts of the regulation of the expression of the lactose operon. There's more to it. There's some, equally cool things about what's going on. But in a word, what I've described to you just now for the regulation of the lactose operon, we call that negative regulation. When the protein that does the regulating, when PLAC-I binds to the operator, it inhibits, it shuts down the activity of an RNA polymerase. It's a downer, right? It prevents RNA polymerase from doing its thing. That's the essence of negative regulation. Since I've told you now about negative regulation, you must be thinking, right? If there's negative, there's going to be another shoe that drops. There must also be positive regulation. Sometimes we find that when we look at an E. coli or a bacterial operon, we'll find that the protein that binds to the operator actually helps the RNA polymerase initiate transcription. Okay? So there's negative regulation and there's positive regulation. PLAC-I is unequivocally a negative regulator. Here's one other thing that's really cool about what's going on with the LAC operon, the regulation of gene expression there. Notice the lay of the land. You got the RNA polymerase that can bind to the promoter sequences, okay? And the way they've got it mapped out here is pretty, true to life. The operator sequence is separate and a little bit downstream from where the promoter sequence is. You know what that allows? What that allows is for PLAC-I to be bound to the operator at the same time that RNA polymerase is bound to the promoter. And the beauty of that, again, this is a testament to how keen bacteria are on getting energy and getting as much of it and as fast of it and as efficiently as they can. Because the RNA polymerase can be bound to the promoter at the same time that PLAC-I is bound to the operator, the instant, not the second, the instant that lactose tickles PLAC-I and causes that allosteric change that makes it no longer interested in binding to the operator, the instant lactose shows up and PLAC-I comes off, you're off to the races and you're transcribing the lac operon. You don't even have to wait for the RNA polymerase to scan along and find the promoter. It's been sitting there all along, ready to go as soon as lactose shows up. Fancy genetics talk for that is the RNA polymerase is poised for transcription. Right word for it, right? It's sitting there, it's poised, it's waiting. Oh, if only I could get past that silly PLAC-I protein, I'd be transcribing away. RNA polymerases are fiendishly fast, hundreds of nucleotides per second, right? So the instant lactose comes off, PLAC-I comes off, bam, you start transcribing that. And remember, in E. coli and other bacteria, transcription and translation happen at the same time. So pretty much just within a second or two of PLAC-I coming off, not only have you started to make an RNA, you've started to translate that RNA and make PLAC-Z because you want to start breaking down that lactose into glucose and get it through glycolysis as fast as you can. And part of what makes this amazingly impressive to me is I cannot begin to imagine a better way to design this system. It's simple, it's direct, it's foolproof, you can't mess it up, right? It's an on-off switch, it's directly responsive to the thing. I might have been able to come up with this if you set me up in a room and I didn't know anything at all about molecular biology and you asked me to design a system with this, I might be able to come up with it. I think you guys might be able to come up with it too. It might take us hours, it might take us weeks, it might take us a couple months to work out something as pretty as this and then to throw in a little twist at the end about poised for transcription. But boy, you gotta just take your hat off and say, nice piece of work, right? About as well engineered, as well designed as you could ever imagine it to be. How are you going to improve on it? I don't think it can be improved on. I've never heard of anybody who's been able to come up with a better way to get an E. coli to respond to the presence or absence of lactose. Make up whatever protein you like. Have it do whatever sort of magic tricks you like. It's going to be hard to come up with something better than what PLAC-I does. So here's the whole picture. Supposed to just to focus on the operator that we were doing before. Here is the inducer. It's effectively lactose. Check it out. When lactose binds to PLAC-I, there is that allosteric effect. It changes the shape of PLAC-I. This thing can't stick into that thing. It can no longer recognize the nucleotide sequences of the operator, and transcription can occur. Whereas this thing could go in and block transcription. So here are the two alternatives. Here we've got PLAC-I in the absence of lactose, so that it is acting as a roadblock. Here we have PLAC-I with lactose present, and transcription is occurring. So pictorially, it's the same thing we've just been talking about. All right. So I'm not at the end of the lecture, but I'm mixing things up on you a little bit today in a couple of ways. The question that I'm going to put on the next test is this one here. I'm going to make a multiple choice question out of this. But if you can navigate your way through this question and the answers, incidentally, this PowerPoint slide is available on Pilot, so it's there for you whenever you like. I'm going to ask you a question like this. If you can wrap your head around this question, excuse me, and come up with ways to get that to be part of the question, I think you're going to be in good shape. So let me see exactly what I'm asking you here. So I've got cells here, 4 different kinds of cells that have different mutations. So I've messed something up. Which of these mutations is going to make it impossible for the cell to metabolize lactose? A mutation in the LAC-Z gene, right? Coding for PLAC-Z or beta-galactosidase. All right. I'm wondering now what my other choices are going to be. It's been a while since I've looked at this slide, but I'm wondering what my other choices are going to be because it's going to be hard to beat this one, right? I'm telling you, I've messed up PLAC-Z. What that means in simple English is PLAC-Z doesn't work. It's broken. It can't metabolize lactose. It can't break lactose into its 2-component monosaccharides. The question is, which of these is going to make it impossible to metabolize lactose? That's a winner, right? So the first choice looks like a pretty good one to me. Let's see what these other choices are. Here I've messed up PLAC-I. Right? So I can't make the protein that acts as a negative regulator, and specifically this mutant version can't bind to the operator. What happens if PLAC-I isn't bound to the operator? What happens is the RNA polymerase just blows right past the operator and transcribes through the LAC operon. You're going to be making a lot of beta-galactosidase and PLAC-Y and PLAC-A. That's not going to stop you from metabolizing lactose. There may not be any lactose around. It's a problem for the cell, but it's not going to stop you from metabolizing it, so I don't think this is a very good choice. What if we mess up the operator so that PLAC-I can't bind to the operator, just doesn't recognize it? It's not what it's looking for. If PLAC-I is not bound to the operator, RNA polymerase is going to blow right by. It's going to transcribe. Okay, that's not going to stop you from metabolizing lactose. It's going to cause a problem for the cell, but it's not going to stop you from metabolizing lactose. And what if we prevent PLAC-I? We have a different mutation of PLAC-I. This one prevents it from binding to lactose. If PLAC-I can't bind to lactose, PLAC-I, it's a 2-trick pony, right? I've just taken away one of its 2 tricks. It's only got 1 trick to do. That's all it's going to do. Its other trick is stick to the operator. If it sticks to the operator, RNA polymerase can't get by. Oh, wait, I can't transcribe the LAC operon. I can't metabolize lactose. So I actually got 2 right answers amongst those 4, right? Both the first and the fourth. So if you can wrestle your way through these kinds of questions, I think you got the LAC operon down. But I want to encourage you, I've given you some tools. I gave you the tool right now in a toolbox of negative regulation. Wrap your head around what are the clever ways that you can use that tool. How can you use that tool to your own ends, to design things that do certain things in response to other ways, and then how do you break it? That's the way a lot of biologists think and engineers think about these things, is what do you do to break this? And then if you change one part, how do things work differently? All right, so there's negative regulation. Let me tell you the rest of the story of the lactose operon. So there's a positive regulator involved with lactose metabolism as well. PLAC-I is a negative regulator, and it's the thing that keys the cell into the presence or absence of lactose. Obviously, I hope, an important consideration when it comes to the metabolism of lactose. Is there lactose present or not? I hope, obviously, an important consideration. There's another thing, though, that E. coli needs to consider when it's deciding if it wants to express the lactose operon. What could that be, right? Isn't it all about lactose? Well, you know what? There's something more important than lactose. The something more important than lactose is the thing that I've been trying to shout at you without hurting your ears. It's all about energy. That's job number 1. Lactose is cool only because it helps you get energy, right? Priority number 1 is energy. Check it out. If E. coli has lots of energy in hand right now, it doesn't need lactose. It's doing just fine, thank you, with whatever else is going on, right? And so why would you spend the hundreds of thousands of ATPs, tens of thousands at least, to make the infrastructure to let you get lactose if you're already swimming in ATP? If you got a lot of ATP in hand, let the other suckers burn some of it to make the proteins that let you metabolize lactose. I'll get around to the lactose after I'm done eating all of the pick your favorite food and insert it there, right? I don't know, cotton candy. I'm fond of cotton candy. I'm inclined to say beer, but it would be more easier for, I like cotton candy. You know, as long as I got a big bowl full of cotton candy in front of me, you know, I appreciate the broccoli over there. It's very nutritious. It has some neat things going on. And I kind of like broccoli, too. I'm a little bit unusual that way. But if you give me the choice between cotton candy and broccoli, I'll go with the cotton candy. When I'm done with the cotton candy, then I'll start thinking about the broccoli. You guys want to go at the broccoli first? You just help yourself, right? Go right ahead. Eat as much broccoli as you like. I'm going to focus my attention on the cotton candy, right? When I'm done with the cotton candy, if there's some broccoli left, okay, I'll think about it. Maybe then I'll start working on the broccoli. But as long as there's cotton candy, why am I messing with the broccoli? From E. coli's perspective, it comes down to this. You know, lactose is a disaccharide. One of the sugars is glucose. The other, off the top of my head, I'm drawing a blank, but it's not glucose.
Lactose?
It could be. It's not glucose. Is the key. And because it's not glucose, guess what? You got to convert that other monosaccharide into glucose to run it through glycolysis. It costs energy to convert that monosaccharide into glucose to run it through glycolysis, so it's not as good a deal as just straight freebasing off of glucose. If you got glucose, just use the glucose. Don't mess around with converting other things to glucose until you're out of glucose. All right? And so, the other thing that E. coli and bacteria in general need to keep tabs on when deciding if they want to express the lactose operon is the energy levels within the cell. If there's already a lot of ATP, don't bother. We're fine. A positive regulator is what communicates that to the RNA polymerase. The way the positive regulator works is this. The positive regulator is cyclic AMP receptor protein, CAP. Okay? You only get cyclic AMP inside of a cell if you've got a lot of AMP, adenosine monophosphate, and you only have AMP if you've already burned through your ATP and converted it to ADP, and you even start to burn through your ADP to make it AMP. Right? Energy levels are getting kind of chancy, you're getting kind of low when you've got cyclic AMP. There's a protein that binds cyclic AMP, and it is also a two-trick pony. But this is a two-trick pony that does both tricks at the same time. I don't know. I got to work on my pony analogies here. But what cyclic AMP receptor proteins does is this. If cyclic AMP is present, it binds to cyclic AMP receptor protein. Cyclic AMP receptor protein with cyclic AMP bound to it is in a conformation that makes it want to stick to the promoter of the lactose operon. Okay? So cyclic AMP receptor protein with cyclic AMP has an affinity for the promoter of the lactose operon. When cyclic AMP receptor protein is bound to the promoter of the lactose operon, the promoter of the lactose operon is suddenly much more interesting to RNA polymerases than it would have been if that protein wasn't there. Molecular geneticists have a word for promoters like the promoter of the lactose operon. It's a sucky promoter. Okay? You heard me, I said sucky. It's a bad promoter. I told you that RNA polymerase looks for about a dozen nucleotides to sit down on. The promoter for the lactose operon has only got 9 of those 12. Yeah, it's pretty far off, right? What the RNA polymerase is really looking for. But you bring cyclic AMP receptor protein into the party, have it bind to that promoter, and suddenly, this is a much more interesting party. And the RNA polymerase has an interest in binding there, whereas it was less interested to do so before because it's such a weak promoter. That's the technical term. Sucky is a word that I use. The word you'll see in the literature is weak. It's a weak promoter. Right? And so cyclic AMP receptor protein is a positive regulator. It only shows up when ATP levels are low. When ATP levels are low and when lactose levels are high, that's when it's a great idea to express the genes of the lactose operon. When either of those two things are not the case, when ATP levels are high, don't bother. When lactose levels are low, don't bother. But when you have both low ATP and high lactose, that's when you express those genes. Negative regulator, positive regulator, that's how E. coli regulates the expression of the lactose operon. There's actually 2 switches that are on, 2 on-off switches, in a sense, that are in play. So here they're showing you a picture of how about how cyclic AMP receptor protein can bind. RNA polymerase likes to butt up against it, feels real good to rub its backside up against the cyclic AMP receptor protein when it's bound. Again, it makes it a more appealing party. The RNA polymerase is sitting there saying, I'd like hanging out with you. And transcription can happen if there's nothing blocking the way for the RNA polymerase. All right, so let me give you just one more example, and we'll call it a day. I've talked with you at the beginning of class a little bit. I mentioned tryptophan operon. Tryptophan's an amino acid. E. coli can make tryptophan from scratch. To do it, it needs to express 5 genes, cleverly named TRIP A, B, C, D, and E. 5-step process to be able to convert unimportant things into tryptophan. Okay? Metabolic pathway. When do you want to express the tryptophan operon? Pose a question to you. What do you suppose is an important consideration? How about, let's only express this operon when we need tryptophan. Okay? When do you need tryptophan? Well, when your tryptophan levels are low, it's a pretty good sign that you need tryptophan. You need tryptophan kind of all the time to be able to make proteins. Tryptophan's one of the 20 amino acids. If you don't have one of the 20 amino acids, you can't really finish making your proteins. They stop, the ribosomes stop whenever they get to a tryptophan codon, and that's not good for anything. So if you're low on tryptophan, yeah, you should probably make some more tryptophan. Good idea, good time. When you already got a lot of tryptophan, it's a waste of your time, a waste of your energy to make the proteins that make tryptophan. But if you don't have tryptophan, you need tryptophan, it's a good idea for you to make those proteins. Simple as that. So how are you going to get that job done? Right? I could leave you just to ponder this, right? But I'll guide you through this one last one, and then I'll let you ponder some others. How are you going to decide? How are you going to regulate the leucine operon? How are you going to regulate, the other amino acid biosynthetic operons? Here's how the tryptophan operon works. There's a promoter, right? There's a stretch of DNA that codes for a polycistronic message, just like you saw in the lactose operon. You got an operator. The operator is something to which a regulatory protein called P-trip-R can bind. When tryptophan is present, it binds to P-trip-R, it changes its shape, and it allows it to sit down on the operator. When tryptophan is absent, I think you'll see what's going to happen is it doesn't bind to P-trip-R, and P-trip-R can't bind to the operator. What kind of regulator is P-trip-R? Positive or negative? What do you think? I'll go with negative. Right? It's a negative regulator because when it binds to the operator, it stops RNA polymerase from doing its thing. cyclic AMP receptor protein is a positive regulator because when it binds to the promoter, the operator of the lac operon, it helps RNA polymerase do its thing. But P-trip-R, solid example of negative regulation. Do you see the little bit of a twist to what's happened here? The lac operon is a catabolic operon. It's breaking things down. The trip operon is an anabolic operon. It's building things up. That has an interesting implication for how the negative regulator needs to work. For the lac operon, you want P-lac-I sitting there when lactose is not around. For the trip operon, you want P-trip-R sitting there when tryptophan is around. And it all comes down to the directionality. One's catabolic, breaking things down. The other's anabolic, building things up. Right? So it makes sense then for P-trip-R to work in this particular way when it works as a negative regulator. All right? And that's my story. And I tell you what, I did finish 3 minutes early, 2 perhaps. So have a great Thanksgiving, guys. I'll see you a week from today. And I'll ratchet it up a little and we'll talk about eukaryotic regulation of gene expression. Similar, but a whole lot more complicated. So anyway, hope you have a great Thanksgiving. See you next week.